Good evening, everyone. Ladies and gentlemen, as President of the Royal Society of Edinburgh, my name is John Arbuthnot. Can I welcome you most warmly this evening to this, what is going to be a really interesting evening. And I know that, having spoken to people who are going to speak. We are gathered here this evening to talk about to have the discussion around Lost at Sea, the Atlantic Salmon's Ocean Odyssey. And having looked at the contributions and looked at some of the background to this talk, it's a really remarkably interesting area. How can a creature this size, at birth, leave its birthplace, travel thousands of miles across oceans, and return to spawn in the same river? And that's what the story is, and you'll hear that that's not an easy thing for these little creatures to do. The Royal Society of Edinburgh, just to tell you briefly a little more about it, not at any length, 1783 was when it was formed by the order of King George III, and it was established in a quite remarkably short way. First of all, it was done very quickly, and secondly, the specification for what this society would do is really brief. It's about the discovery and development of new knowledge and the useful application of that knowledge. That's it. And out of that has grown a society which covers every discipline that you can think of, from science to medicine, to law, to ecology, to theology. 2 weeks ago, we had the previous Archbishop of Canterbury here talking about the theory and principles of theology. Every discipline is covered by fellows of the Royal Society of Edinburgh, and that's a source of great inspiration and pride for us. So that's the background to the society. I welcome you here. It's a sellout audience, and we have people in the overflow as well. And I'm now going to hand you over to our chairman for this evening and to ask him to take us through the business. And that, of course, is Professor Michael Usher. And over to you, Michael.
Thank you, President. Good evening, ladies and gentlemen. I should like to add the welcome of the organising committee as well. That's Lord Nixon, Dr. John Baxter, and Dr. John Hutchinson. I want, in introducing this evening, just to make 2 points. The first is that it is a real pleasure to see so many people interested in the salmon and its migration. Although the salmon occurs more widely, it is in fact one of those iconic species for Scotland. It is also one of the rather few wild living species in Scotland that is economically important. My second point is that salmon features in a publication from the Scottish Government just 3 months ago in August entitled Key Scottish Environment Statistics 2013. There's an interesting graph in that publication which looks at the year-to-year catches of salmon over the last 60 years, from 1952 to 2012. During this time, the rod and line catches have remained more or less constant at something like 60 to 70,000 salmon per year. However, something like 400,000 salmon were being caught each year by fixed engine, net and cobble in the 1960s and early 1970s. In the last few years, that number, 400,000, has dropped to something like only 20,000, a 20-fold decrease in the number being caught. With such a large decline, I think we can ask the question, where have all the salmon gone? And with that, I would like to introduce you to Dr. Peter Hutchinson, who's going to introduce this evening's topic for us. Peter.
Well, thank you, Michael. Good evening, ladies and gentlemen. Who'll define a salmon? The riches of this fish could make all tourist boards rejoice. He's granted their utmost wish. His wisdom solved the riddle of the famine in the dish. These are the words of Ted Hughes, a former poet laureate, about the subject of this evening's discussion forum, the king of fish, the Atlantic salmon. Annual migrations involving concentrations of individuals in space and time have long fascinated mankind and represent a readily harvestable source of food, at least some of them do. Constraints on production in fresh water mean that Atlantic salmon is not as abundant as Pacific salmon or marine fish such as the Atlantic herring. The peak catch for the whole of the North Atlantic was around 12,500 tonnes, but today catches are only 1/10 of that. However, this species is highly prized and creates economic benefits wherever it occurs. For example, expenditure by anglers here in Scotland has been estimated to be about £73 million annually. But that's only one component of the salmon's value. People care about this species, as the attendance here tonight demonstrates. It is seen as an indicator of a healthy aquatic environment, and people are intrigued by its remarkable life cycle and marvel at its acrobatic displays as it migrates upriver to spawn. In short, people are willing to pay to conserve the salmon, irrespective of whether or not they wish to fish for it. A recent study in England and Wales estimated that the overall economic loss to society from a severe decline in salmon stocks would be £350 million per annum. So it's a small but highly valuable resource. Now, because of its migratory life cycle, the salmon is a truly international species. After a variable period in fresh water, up to 8 years in the north of its range, salmon undergo a remarkable transformation. They become smolts, an adaptation that prepares them for the very different marine environment they will encounter during their ocean life. This journey may take them from their river of birth to the northern Norwegian Sea and even as far as West Greenland, and then precisely back to spawn. This marine phase may last from 1 to 4 years. In the rich marine waters, salmon grow quickly on a diet of fish and crustaceans, but the ocean also poses different challenges. In the 1960s and 1970s, fisheries developed in the salmon's feeding areas at West Greenland, in the northern Norwegian Sea, and around the Faroe Islands. That is one reason why there is a need to manage the resource internationally, and it is why the Atlantic salmon is one of a small group of fish with an international treaty dedicated to its conservation and rational management. This treaty created an enormous sanctuary, free of salmon fisheries, covering most of the North Atlantic. It provided a mechanism to regulate certain interception fisheries, and it provided a vital international forum for cooperation on conservation and acquisition of scientific information. 30 years ago, catches in the distant water interception fisheries were the burning issue. It was believed by some that if these fisheries were regulated, salmon abundance would rebound. All would be well. That proved to be too simplistic a vision, focusing as it did only on a single issue, important as it was and still is to limit those fisheries. Through international negotiations run here from in Edinburgh, those harvests have declined to the extent that the combined catch has fallen from around 3,000 tonnes in the early 1970s to an average of only 22 tonnes in the last 10 years. Huge sacrifices for countries dependent on marine resources. There have also been large reductions in harvests in the home water fisheries. But despite all these sacrifices, the abundance of salmon prior to any fisheries has declined markedly and remains low. Mortality at sea has increased, linked, it seems, to changing ocean climate. The decline is most marked for southern stocks and for those fish that spend more than one winter at sea. These graphs show the trends for 2 of the stock complexes that scientific advice is provided for. But I should say these are stock complexes representing a large number of rivers that will be performing differently, but they do highlight the overall situation. The problem is very clearly illustrated here for just one river, the River Bush in Northern Ireland, part of a network of scientifically monitored rivers where the adults are counted in and the smolts are counted out. You can see that despite an increase in the number of smolts, the dashed line on the graph, the returns of adult salmon after 1 year at sea, the solid line, have declined sharply. Additionally, as we will hear shortly this evening, in some areas the condition of returning salmon has also declined. Now, until recently, relatively limited research had been conducted on salmon at sea, and much of what we did know was obtained from sampling the catches in the distant water fisheries. The challenges facing scientists looking into this issue were large, but there has been real progress, including through an internationally coordinated programme of research known as SALSI, which we'll hear more about shortly. New insights have been obtained into the salmon's ocean migrations and distribution, its diet and growth rate, and new tools to support management have been developed. While management options in the open ocean are limited, there are emerging concerns about bycatch in the large-scale pelagic fisheries. This slide shows that the catches of mackerel, while research fishing for salmon, can be high. We know also that salmon are being caught in commercial fisheries for mackerel, but estimates are highly uncertain. The SALSI programme findings should facilitate better quantification of this and inform the need for measures to protect wild salmon. What we can influence, however, is factors operating in fresh estuarine and coastal waters. And as we'll hear again later on from one of our speakers, new challenges are emerging in these areas close to home. To quote one salmon conservationist, "Mother Nature has thrown down the gauntlet. She must not be allowed to throw in the towel." I'm sure that our three speakers tonight, all internationally respected salmon biologists, will give us a marvellous insight into how recent scientific endeavours have started to unravel some of the mysteries of the salmon's incredible ocean odyssey and how we can use this information obtained to support better management. Thank you.
Thank you, Peter. Can I introduce now Ken Wellen from the Atlantic Salmon Trust, who's going to talk about unravelling the problems of the salmon at sea and the major SALSI project. Ken.
Thank you, Michael. Good evening, ladies and gentlemen. Peter has done a very fine job in setting the scene for me to talk a little bit about our Salmon at Sea project and to bring you up to date in terms of some of the results that we have found. Certainly, at this stage, I think it's fair to say that the greatest challenge that we face in terms of salmon conservation is to gain an insight into the spatial and ecological use of the marine environment. Remember that this challenge faced us around 2002 in a situation where the vast bulk of scientists working on salmon had training in freshwater, not training in terms of the marine environment. As Peter has shown with the two graphs, this is the situation that we faced in terms of 2005. And that PFA at the top of the first slide, that's pre-fishery abundance. So in simple terms, that's an estimate of how many salmon were mustering off the coast before they actually returned back to their home waters. And as you can see, a very dramatic decline in terms of the numbers of salmon. Much more so, as you've seen in the previous graph, in terms of the southern stocks than we were seeing in terms of the northern stocks. So immediately we were beginning to see a pattern whereby the stocks in the south were doing much more poorly than the stocks in the north. It was a matter then of trying to pull together partners who would actually tackle this particular problem. And certainly it was a major issue both in terms of logistics and in terms of funding. This just gives a summary of the partners that came together on the European side of the programme. As you can see, 20 separate partners spanning a whole range of different interest groups, from private sector groups such as the Atlantic Salmon Trust and the Total Foundation, to the research bodies in the various countries, and also to individual universities as well. Remember in 2002, when we started to discuss this, we hadn't got a single penny. And we were facing into a situation, we realised within about 12 months of discussion, a situation where we were really looking at a programme that was going to cost in the region of £17 million. That programme needed to span a whole range of different areas, right across that map that Peter showed you earlier of the North Atlantic. We were very fortunate in the context of the European component of the programme to be able to get some funding from the EU through the FP7, the framework programme, 3.5 million. And onto that then was bolted all sorts of different supports. And we ended up in a situation where we had programs that ran right across the area that was important to us. So we had programs running from West Greenland, West Iceland, the area that was in the east, if you like, the east coast along the eastern margins, if you like, of the salmon's distribution along the west coast of Norway and up into the Barents Sea. So we were very fortunate in being in a position where we could then start to assign specific areas that we were going to survey. And we were going to look into these areas and we were going to try and figure out where the fish were going and how they were getting there. Now, there is some confusion, I think, in relation to the first phase of the SALSEA programme, because the real issue was the issue of why fish were dying at sea. This wasn't our primary objective in phase 1. We needed to try and discern the migration and distribution. We needed to know where these fish were going, and we needed to know the pattern of movement, not just of salmon per se, but of individual stocks. We needed a method where each little salmon smolt or post-smolt that we caught at sea, that each of those fish was able to tell the story in terms of the stock that it came from. And we managed to do that with some very innovative genetics, basically CSI salmon. The same technology that you see every night on television, cracking crime in North America, was the technology that we used to try and individually identify the stocks of fish. The other problem that we had, and this is a very personal slide for me because this is the Celtic Explorer, one of the ships that was belonging to the Marine Institute, a group I used to work for. I had to go before the board of the Marine Institute some years ago and explain to them that I wanted their ship at something like €18,000 a day, that was 65.5 metres long, and I was going to chase these little fish that were 12 centimetres long. I had no idea where they were, I had no idea where they were going, but I was going to find them. And foolishly, they agreed. My colleagues in Norway learnt the lessons from me, and they found the same reaction. People were fascinated with the idea that you could actually go to sea, that you could put on a map a set of lines, and that you could build from those lines then a migration and distribution pattern. So that just gives you an idea of the sorts of tracks that we handed to the captains of the ships. And after the first year working on this particular programme, we realised that the gaps, and they are quite substantial gaps between the lines, that really we needed to start to move up through the lines. We needed to be sampling not just in this very symmetrical pattern, but do something a little different. And we sent the Celtic Explorer that you saw earlier to try and weave its way through the original tracks. That was a very good decision because we actually located in some of those areas where there was clear evidence of smolts that were doing very poorly. There were what you might call black zones. And as Peter has mentioned, the technology that we put in place to actually do this, certainly for simple freshwater biologists like myself, was awe-inspiring. The idea that we could put out to sea these massive great nets that were floating in the surface of the ocean, that we could gather in all sorts of different collections of sea fish, and that amongst those we would consistently find salmon smolts, was actually an extraordinary success in itself, and really down to the technicians that worked with us. We soon found out that using nets was quite damaging in terms of the samples we were trying to collect. It was descaling them and so on. And the Norwegians again came up with a very clever box system. We were actually catching the fish alive in the box. And it was extraordinary that in terms of all of the surveys that we carried out over a 2-year period, something like 97% of the hauls had salmon included in them, which I think is testament to some of the earlier work that was done by Dr. Dick Shelton and by Jens Christian Holtz in the late 90s. So what we ended up with then was an array of dots, all sorts of information that was collected in the context of this particular study and information that was collected previously. And we soon learned that the distribution pattern of these fish was, as we had hoped, really quite specific. We also learned very quickly that not alone were we unearthing tags for individual river stocks of fish, but there were patterns that were showing us regional tags as well. Groups of fish that after the Ice Age had come in together had retained characteristics that were even today discernible by genetics. And certainly it was clear that the distribution of the fish was linked to ocean currents. And as Chris will tell us in a moment, the whole issue of the food supply in the ocean that again is intimately linked with that question of temperature. It was also interesting to see that in terms of the pattern that emerged, this fish is so variable, even though the generality of the graph that we saw earlier is definitely a trend downwards. During the period that we looked at, there were years when the fish did relatively well, but there were also years when the fish did really, really poorly. And I think that was really support for what we had found in the cruise, where we found these spots, these locations in the ocean, where there was particularly poor feeding for these fish. The other fascinating finding, and we, again, thinking about this just theoretically before we went to sea, had imagined that the great ocean currents were the defining currents, the currents that were driving forward up through the ocean. But in essence, what we found was that wind fields were so important that the actual migration routes were very, very much influenced by the currents in the ocean layers at the very surface of the ocean. But thankfully, we were in a position where we could start to build ocean corridors. And to me, this is one of the major breakthroughs. The idea that we can now not alone look at a scientific area of conservation that ends in an estuary, but that we can spread it out. We can move along the ocean. Again, as Peter was showing us, there's a huge overlap here. There's an overlap between the migration routes of the salmon and, as you see, the areas where we fish for mackerel and herring and blue whiting that are in our fishmongers. So we need to be aware of this. And recently we've been meeting with the skippers who actually run the pelagic fleets in these areas, giving them an idea of the maps that we have constructed that teaches us exactly where these fish are located. There's a huge wealth of information available on all of the survey work that was done over the last 10 years, and it's readily available on the web, and this information I'm sure will be available through the Society, and you can click into those links and learn a little more. But finally, just to look at the future, let's just look at phase 2. So in terms of phase 2, what we're looking at is a situation where we really have to tackle this whole question of where the fish are dying and exactly what is causing the death of these fish. We have decided again, through Peter's good offices in NASCO, to be even more ambitious, and we have decided to partition the ocean. We have decided to start looking at modern technologies, to look at the points in the distribution of these fish at sea and their migration routes, the areas where they change current, the areas where the currents are at their narrowest. There's all sorts of fascinating technology. This will bring us all back to putting those little ships on the ponds in our local park. But these little ships are the most amazing little pieces of technology. These little guys can go out and use the waves themselves to energize and to move across the oceans, picking up the tags from the fish that we are going to release. Already through the Ocean Tracking Network in Canada, there's a battery, an array of receivers at sea that we're going to try and capitalize on. And in essence, we need to move away from a paradigm which we've had for over 100 years, where we always saw survival of fish and monitoring of fish from the estuary inland. And we now have to look at something which is all embracing a vision of salmon populations which encompasses survival corridors. We're back to these corridors again. And these corridors, remember, stretch from the remotest spawning burn in the Scottish Highlands to the very limits of the salmon's migration. That's a great concept, and I think it's one that really should give us great encouragement to tackle these things. So finally, what is my wish list? Well, basically, as I say, the new programme, I would love to see that brought to fruition, to understand exactly why and where these fish are dying at sea. I would like to see a situation where our masters in Brussels think again about this designation of scientific areas of conservation. Why can't we have it stretching out into the oceans? Why can't we have a marine protected area that actually protects individual stocks at sea? We need to quantify and tackle the suggested impacts of the pelagic catch. And the captains themselves are interested in doing that. And certainly we have missed a trick, ladies and gentlemen. We are now almost 4 years since we were last at sea in terms of SALC. We haven't taken a single sample at sea. And I feel we are too complacent. I think we need to be out there. We need to be gathering these fish and looking at least at the growth and condition of these fish, which would give us a very good indication in terms of what individual year classes were suffering at sea and what we might expect then in subsequent years. Thank you all very much.
We have a few moments if there are one or two immediate questions. Of course, there'll be a more general discussion later after the three speakers have spoken. But are there any questions just at the moment? Jack Matthews. Sorry, Jack, can you just wait for a microphone so that everybody can hear? One's coming the other way.
Hello, Jack Matthews, formerly of SAMS. I'd be interested to know whether you've touched base with people in the North Pacific, where the position with salmon, of course, is parallel but very different because you've got a multi-species situation. The Canadians in the North Pacific have worked quite a lot at sea, and they've worked with planktologists and the food for the salmon in many cases. I wonder whether you're touching base there, and clearly the questions you'll be asking will be parallel, but the cooperation there, I've had a lot of experience in Norway, and we worked there from the fjords out into the sea, working with the plankton and to some extent with the fish. The Sir Alistair Hardy Foundation for Ocean Science has worked with the Continuous Plankton Recorder for years, and they've got an amazing set of data for looking at the food supply of the fish. I'd like to see what is the coordination going on between yourselves and the very interesting work you're giving and these others. Thank you.
Thank you very much indeed for the question, an important question. The ICES Journal of Marine Science that was published in 2012 has a very good collection of scientific papers, including papers from the Sir Alistair Hardy Foundation, which indicates very clearly that we have a real serious problem in terms of the North Atlantic, not just in terms of salmon, but in terms of the planktonic component of the sea. We have worked very closely with our colleagues in North America since the programme began, and indeed, again, they were present at our Salmon Summit some years ago. David Welsh, who was involved with the original POST programme, actually gave us the idea of the original SALSES concept. So it really started with that particular initiative. So yes, we're working quite closely with them. But interestingly, they are finding quite contrasting results in terms of what's happening in the Pacific versus the Atlantic. So that work certainly needs to be intensified, I agree.
There's a question down in the front here. Oh, sorry. And then.
Yes, Eric Salzon. Has there been a correlation over the years between the decline in salmon and the increase in farming of salmon?
I think that's a question maybe we might ask Chris about as well during the general discussion. My understanding is at this stage there have been several papers where there have been some correlations claimed in relation to that. My own view is I still think that it's an area where it's unproven at this particular point in time. If you have a situation where in the remotest areas of Labrador, where the rivers are so remote, they have native Indian names, but mostly they're numbered. If you're seeing a similar decline in those situations as you are in areas where there are much greater populations of man, there's definitely an underlying marine problem. That's not to say that the other problems, whether it's fish farming, whether it's problems in freshwater or whatever, that they're not serious. But I think the underlying problem is the one we touched on earlier. It's the climate change problem, it's the changing oceans. That would be my view.
There's a question back there. Sorry. Was there a question there? No. No, sorry, I thought you were... Ken, have you managed to persuade the European Commission that they need to relook at the marine side of the Habitats Directive with your marine protected areas and corridors?
We had two very good meetings with both DG Environment and DG Mara. So we've certainly planted the seeds. Now, the reaction was really quite predictable in the sense that any good civil servant will really balk at the idea of changing legislation. But they did accept the fact, while they couldn't extend out the SAC concept, they did accept the fact that in terms of marine protected areas and the Marine Strategy Framework Directive, that it needs to be seriously looked at. And I think it's the way rather that we have to move. We must move in that direction. And I think it's going to take increasing pressure. I must say, and I'll repeat myself, I do think that we're missing a trick here, in that if we keep leaving gaps in terms of our knowledge base, we had this great bit of excitement over a 3 or 4 year period. We were collecting the information, but we have now lost almost 4 years. That's a full cycle. That would give us a full grilse, if you like, in terms of egg to adult. And we can't afford to do that. We need to convince the powers that be that if we're investing taxpayers' money in terms of surveying the oceans and monitoring the oceans for all sorts of reasons in the context of EU agreements, salmon must be included as part of that pelagic package. I think that's really important.
Thank you very much indeed. We need to move on. If I could now introduce Chris Todd from the University of St Andrews, who's going to be talking about the importance of data for salmon fisheries. Chris.
Good evening, ladies and gentlemen. And firstly, could I say thank you, sir, to the Royal Society for the opportunity of speaking to you. And also, before I forget, to say thank you to Orla and Roshin and also Brian for all the help you've given me in getting myself to the state where I can actually talk to you about dates with salmon at sea. Now, dates mean different things to different people. And what I'm going to do is to take you part of the way along the journey. I'm not going to show you the final destination. But also, right from the outset, I wanted to thank these various institutions who, over recent years, have contributed to maintaining some of the monitoring of wild salmon that we've been undertaking here in Scotland. So thank you to those for that assistance. Now, Peter and Ken showed you the pre-fisheries abundance data that are computer modelled by the North Atlantic Salmon Working Group. These are the official ICES data for nominal catches of salmon. And in essence, you can see a very broad pattern from 1960 through to the present of very high numbers, especially in southern Europe, in the turquoise colour here, and very marked declines. Now, I emphasise that these are catch data. These are only a proxy for abundance, but they still give you the broad picture upon which I want you to place the results that I'm going to show you, which is changes in quality or condition of salmon. I'm not going to be talking to you about abundances, but this is the background image upon which we need to actually place this. Also, one other word of warning, some fisheries, as Peter has mentioned, have closed, and so these reductions in abundance in catches are partially responsible for the pattern that you see. But I emphasise I'm talking about quality, not quantity. Now, those of you who are familiar with wild salmon, rather than the rather different fish that we see on the supermarket slab, this is the kind of fish which we would all like to see returning to our rivers. And in this particular case, the individual is about 4% underweight for its length. But for all intents and purposes, we can take this as being a normal fish in terms of its weight at a given length, in terms of the long-term average. Now, this is more like what we've been seeing with increasing frequency in recent years. Now, those of you who will be catching kelts in the river in February, March next year would easily mistake a fish like the top for being a kelt, but this fish has not even left the marine environment. It hasn't yet re-entered the river when we actually intercepted it. Now, two other important points I need to make. The condition factor index is shown as WR. This is a ratio between the observed weight of the fish and the predicted or expected weight that a fish of that length should have. So if it is skinny, it has an index less than 1. If it is fat, it has an index greater than 1. In fact, a normal fish, for reasons I needn't bore you with, a normal fish has an index of about 0.96, 0.97. So the top fish here is about 26% underweight for its length. The other important thing is that I'm only going to be talking to you about one sea winter fish which return to freshwater after about 12 to 18 months at sea. And these fish, we know from tag studies, primarily migrate to the Norwegian Sea, so we can target the area upon which to actually focus our attention. Now, when I first started working with this, I noticed patterns in the apparent changes in quality of the fish. And I'm expressing condition here as a predicted weight at a standard length. in essence, the pattern you see here is exactly the same if you calculate condition as that WR index that I've just introduced you to. So for all intents and purposes, we can take this as being a comparable measure. Now, the first instance we had for this was from monitoring commercial catches at Strathy Point on the north coast of Scotland. And the important feature here is that this is a mixed stock fishery. It's not associated with a particular river. We do not know the river from which these fish are actually derived. Now, Ken's genetic typing would allow us to actually identify those river stocks. But one thing which became obvious was that we saw this pattern of an early high level of predicted weight falling off as the turn of the millennium proceeded. And it was distinctly possible that that fall in weight was simply because I happened in the later years to be measuring or sampling fish from a river where they typically are long and thin. So the River Spey, for example's fish are commonly this beautiful slim torpedo shape, whereas if you go a little further north on the east coast to the River Brora, for example, you will typically see very short, deep-bodied fish. So it's possible that this pattern is simply an artifact of biased sampling. Now, fortunately, Marine Scotland have detailed archives for the River North Esk going back to 1963. We looked at those, and in essence, you can see exactly the same pattern. So the cut to the chase, what's driving this? And the answer is that we look to ocean climate change. And I have to emphasize also that I am not inferring a direct effect of temperature on salmon. Rather, it's an indirect effect of temperature shifting the distribution, the availability of prey organisms to this fish. So climate change here is an indirect effect, I believe, on salmon quality, their growth experience at sea. Now, this is where you need to be alert because I'm going to show you 30 years of temperature data in 30 seconds. It's monthly, and if I click on here, we should hopefully, Brian has done his stuff. Right, stop. Thank you, Brian. You're a genius. Okay. This is the northern Norwegian Sea. This is the area that we're interested in. Now, as Ken showed you, salmon are occurring in the eastern half here. They do not occur in the western half of the Norwegian Sea. It is basically too cold for them. I'm going to show you monthly from 1983, 1981, the temperature anomalies. These are the departures from the long-term average for each month over the past 30 years. So if the temperature is a degree or more above normal for that month, it's in dark red. If it's colder, it's in dark blue. So very quickly, are you ready? Come on, play. Please play. What you will see in the first few years, and this is the area of interest here, we're into 1986, really nothing of any particular interest. Up to 1990, still nothing particularly striking. This is 1996, and from now onwards you will see really quite a marked change. This is 2001, 3, 5, 6, 7. And up to April of this year. Now, what you see at the bottom here is a plot of the temperature anomalies for an area of the Norwegian Sea that we know these fish migrate to. And it's not rocket science to see that there is an almost perfect inverse relationship here between those temperature anomalies and the quality of fish that return. I want you also to notice, as you saw in the animation, that in almost every month the temperature since the late 90s has been above the long-term average. But we need to be able to go a little bit deeper into this. And in essence, what we do is we take the annual time series of salmon condition that you've seen, and then we slide that back against every month that the fish are away at sea. So we compare the time series with all the January temperatures, all the February temperatures, all the November temperatures. And the key result that comes out of this is that in January of the year, the same year that the fish actually return, that's after their one winter at sea, there is a very strong negative correlation between the temperature anomaly and the quality of salmon that we see returning to Scotland. Now, in essence, we can see I could see what the temperature is going to be in January, February, when the data are available. we should be able to predict the quality of the fish that will return some 6 months later. So it looks as though the key part of the migratory cycle where condition factor quality is being determined is in January, the beginning of their second growth season. That's an important result which I want you to keep in mind. Question is this different? Is this unusual? Well, again, having privileged access to John Armstrong's data back to 1963, you can see that really between 63 and 93, nothing of any particular interest occurred. In the late 90s, things improved dramatically, and then it fell off a cliff. Now, I remember a few years ago speaking to the Association of Salmon Fishery Boards and saying, look, guys, you could be in for years of skinny salmon. And then on the way home thinking, that was a stupid thing to say, Chris. But actually, I can tell you, I haven't been able to update these, but for 1993, at Armadale, which is a mixed stock fishery on the north coast, the condition factor is 0.9. So it is just bouncing around at this really very low level. And it may stay there for decades. Now, to understand, to conserve, to manage this animal in the future, I really strongly believe we've got to understand the past. And in that sense, we have unique sources of data. But what I've just shown you is simply correlation. And correlation does not mean causation. I can show you very strong patterns or very strong evidence that ocean climate is driving this pattern. But ideally, I want something from the fish themselves. And the fish actually are brilliant data loggers. They record the temperature profile that the fish had throughout its marine life. They record the increase in length. And from the scales particularly, we've for many, many years been able to derive, first of all, the river age of the fish, how old the smolt was when it left the river. We can determine the sea age by simply counting the winter annuli. These are very closely spaced series of rings which are characterized by the winter period. And we can also back calculate the length that the smolt had when it left the river. And that's for one very simple reason, that thankfully salmon don't increase the number of scales as they get bigger, but the scales themselves get bigger. And the scale radius along the horizontal line here is a direct linear relationship. If I know the length of the fish at capture, I can back calculate its length at any ring. But I can tell you nothing about the change in mass of the animal, only its change in length. But it's still powerful information. So, the burning question here is that we could actually gain a great deal more information if we could just simply place a date on these rings. I can back calculate its length at any ring. If I know the date, I can then start calculating the specific growth rate between each of the rings on the scale. So I can reconstruct the growth history of this fish, or each individual fish. Now, specific growth rate, for those of you not familiar with the term, it's how the fish increases in length in centimetres per day. So it's simply how much of a centimetre per day. So just to briefly go back to this, and I'm not going to explain the details, I would be happy to discuss it with you afterwards, but we have actually developed a methodology where we can estimate a date for each circulus. And we can now actually start recognizing when growth hiatuses occur on the scale. So at the bottom here, you have a fish which left the river here and had very fast, steady growth up to its winter annulus, and then grew pretty well before we caught it. Here's a fish at the top which actually grew quite slowly on first entering seawater. It then grew fairly fast, had its winter, and then grew fast again after the winter. The middle here, you have a fish which actually shows what we call a growth check, where the rings are sufficiently close that they look very much like a winter annulus. And these are of particular interest because John Armstrong's colleagues showed that these so-called growth checks were essentially absent from salmon prior to the turn of the millennium. They have increased in frequency in recent years, and that gives me a clue. Perhaps what I'm seeing here is these growth checks underlie the changes in patterns. So what you're going to see in the next few slides are individual fish as rows, and I've dated the growth patterns here. And so slow growth is in cream, fast is in gold, a check is in green, and winter is in blue. And these are the fish, about 600 fish from Armadale, 2010 in sample order. and I think you'll agree it's a bit of a mess and doesn't really tell me a great deal. But if I actually sort them by patterns, a lot of structure suddenly starts to emerge. And we can see that some fish have what I call a slow, fast growth strategy. Others have a slow, fast check. Others had slow, fast check, and then fast growth again prior to the winter. Now, in looking at this, it seems to me, well, maybe what I'm seeing here is this is a group of fish that followed a particular migratory route, as Ken was suggesting. Maybe these fish followed a different route, but they've all encountered, these ones in the middle here, all encountered a growth hiatus in September, October, November time. So that then gives me a clue. Well, maybe we start focusing on those autumn months to try and understand the growth hiatuses. But notice that some fish in gold here grew steadily and showed no problem at all. V is a sort of a dumping ground for anything which I can't categorize, goes down as a variable V. So I expected in the following year to see something completely different. If, for example, how could I guarantee that there was going to be a growth problem or a feeding problem for salmon in October, November? Well, in 2011, we saw broadly similar, slightly different proportions. And in 2012, again, an increased proportion of fast fish, but there's something I don't understand here. I'd be really delighted if somebody could give me a lead here. How is it that 3 years in a row, we're seeing the same patterns, the hiatuses at the same time of year? There's something more than just simply availability of food or fish happening to grow well in certain circumstances, not so well in another. But I've got to remind you that that January temperature result suggested that it was the post-winter temperature which drove the adult condition that I see of the returning fish. And there is no relationship in those first graphs that you've just seen. None of the groupings are particularly high or low condition. They all seem to have a mixture of plump and skinny fish. But I emphasize that I've only shown you pre-winter growth data, and it's the January temperature was indicated by SST to be the critical month. So, back to the North Esk. Now, fortunately, John's people also tag smolts as they leave the river, and in a few cases, we actually managed to recapture them. So in these cases, I know the date that the fish left the river, I know the date that it was caught on, and we can then, from the methodology we apply, we can then date all the rings on the scale. That then allows me to calculate the specific growth rate. This is centimetres increment per centimetre per day in that pre-winter period. And regrettably, in relation to condition factor, there's no relationship. But if we look at the post-winter growth period, we end up with a significant relationship. Fish which have a faster specific growth rate in length tend to come back in higher condition. Now, some of those amongst you who are familiar with quantitative data will say, well, hang on a minute, Chris, it's a pretty scattered relationship you've got there. Two things I'd remind you. The first is that these fish were caught in 4 different years, so they will have had a different growth experience. And secondly, condition factor is a measure of the length of the fish and its mass. We can gain no information on the mass of the fish. We can only gain information from the scales on the length. So actually, I'm not too depressed by that result. But the important thing is to try and see what real data for wild fish tell me. And to cut to the chase, we've looked just at the 1 year. Ellen is here with me. She's helped me measure. We spend hours and hours measuring scales. It drives you mad. We've done hundreds of these for 2010, and you can see exactly the same pattern. And what I want to do now is to actually go back through that wonderful archive that Marine Scotland have. Because if we can understand this, and if we can see the consistency of this pattern, and I have no reason to suspect it won't be consistent, then I think we will be in a much better position to actually understand this fish and its future. So that really is the message I would leave you with. I've taken you so far down the road, I can't give you the final answer, but I think the fish themselves actually contain an enormous amount of information which, with effort, we can glean. I think also we can learn a great deal from the history of this animal as to how to manage and conserve it in the future. I can't tell you how climate change is going to impact this animal. I can tell you that if it continues to warm, then southern Europe and also the United States, those rivers will go extinct. But in Russia and northern Finland, ice-up rivers will suddenly become open to this animal, and so we've already seen that the northern European stocks seem to be faring much, much better than the southern European stocks. So it may well be that things will shift up even further. But Scotland, God willing, will still have this wonderful fish, but it's up to us to actually conserve and manage it. And I think that the history of the records that the fish provide actually gives us a lot of that information. Thank you for listening.
19 minutes. We'll do one question. Sorry. Is there one quick question? Sorry, in the front there.
Stand up.
Thank you for a fascinating presentation. You don't want any new variables. So my question is, with the escapees from fish farms, fish with the different genetic makeup are located in a different place. Is the number of escapees so small that it cannot confound your figures in any way?
All I can say is that all of the monitoring that John's people do with the very best intentions exclude any farm escapees. So the fish that we're looking at are genuinely wild. Whether there has been genetic introgression, breeding of those fish with others, I don't know. But the only other thing I would say, with the exception of the data from Strathy Point and Armidale, which are on the north coast, and we know that from tagging studies that those fisheries sample fish from the west coast, that major pattern I showed you from 63 to 2010 is based, sadly, on east coast rivers, on the Spey, the Tay, the North Est, the Tweed. So in a sense, it's, if you like, remote from the salmon farming regions. So I really wouldn't want to comment on introgression. I'll leave that one to John to answer in the general discussion.
Thank you very much indeed. Thanks. We'll go on then to invite John Armstrong from the Freshwater Fisheries Laboratory of Marine Scotland to talk to us about migration in the coastal waters. John.
Thank you. Thanks, Chris, and thanks for noting the importance of collecting those long-term data sets and how useful they've been to you. We need to keep that going. You mentioned that you had some of my data, Chris, from 1963. Well, I have to say that wasn't my personal data. I was only a toddler then, but and I wasn't very precocious. Coastal migration. Well, we're dealing now with the part of the life cycle. Oops, there we go. The post-smolts and the returning adults. So what are the challenges for these fish? Well, quite major challenges when one reflects. Post-smolts, these small animals need to locate productive northern feeding grounds, as has been expressed by Ken and Chris. No mean feat. And for the adults, having found those feeding grounds, they have to find their way home, in many, many cases, to the very stream, the very area of stream perhaps, where they were themselves spawned. And they're equipped to achieve these tasks with a range of senses, and two very special senses for salmon, an ability probably to detect magnetic fields, to have some sort of a compass, and an extraordinarily well-developed sense of smell. As a smolt leaves the stream, its nose grows enormously, and it is putting down tissue that is encoding smells that it's picking up on the way out. So in the coastal zone, there are a range of challenges, natural challenges, that have been there as long as the salmon, such as seals and other bird predators. But as we start to use the coast more, a range of anthropogenic challenges, challenges from man. And of course, recently, you'll see in the news every day, there's the development of marine renewable energy extraction methods. As has been discussed already, there are problems from climate change. There are problems from fisheries, potentially, and there are potentially problems from fish farms. There are a range of challenges to the fish that are part of the way that we use the planet. So looking at some of the most recent challenges from offshore fish farms, and to give you a bit of background, there are a number of sites now where there are wind farms, and in this case the footprint's quite small. There are some very interesting new devices that will harness wave energy, and you can see the scale of these devices here. And there will be some turbines where a lot of the action is underwater here. So some interesting changes in Scotland's coastline over the next few decades. So thinking particularly about the marine renewable energy impacting factors, potential impacting factors, there's electromagnetic fields from cabling. Of course, if a fish using compasses, these might be relevant. There's a possibility of strike. There's noise during construction operations, and other possible factors such as a build-up of predators around these structures which might tend to aggregate fish. So a lot of new issues to consider. And in relation to a lot of these possible impacting factors, well, what are the key issues for scientists to try and understand? Well, we really need to know about the distribution of salmon in space and time. We need to know where they are in the water column, where they are around the coasts. And we need to know the magnitude of potential impacting factors. How serious might they be? Might they interfere with fisheries? And then moving on further, might they actually interfere with the conservation status of populations? We need to understand about the resilience of populations. So these are key issues that are high on the agenda. Okay, so let's think a bit about the smolt emigration first. And I think as Ken nicely showed, we have a conveyor belt of current moving up. And if smolts can find this conveyor belt, then they're certainly getting a big helping hand to getting to these northern feeding grounds. And the issues that are important here are the currents. Wind can have a big forcing effect, some meteorological conditions, swimming speed of the smolts, and their swimming vector. So partly the smolts will be relying on the natural push, and partly they will be having an influence on what's happening. So for smolts on the west coast and maybe Ireland, finding this conveyor belt is an obvious advantage to them. If we start thinking about the east coast of Scotland, things are a little bit more complex. We have currents that are, to some extent, heading in the wrong direction. We've got gyre systems here, and it's not entirely clear how smolt should behave. How should they best find their way north? Should they hug the coast? Should they move out here? Should they try and find the Norwegian coastline? We just don't know at the moment. But we have started to get some information in the more inshore areas on the west coast. It's possible to put little acoustic transmitters inside salmon smolts. And on Loch Linnhe, looking at the interaction between modelled plumes of lice emanating from fish farms and the distribution of smolts, we've tracked fish using listening stations to pick up these tags. And what we find is that the fish generally move very quickly out to sea through these systems. So they seem to know, well, they seem to have an inbuilt compass, perhaps, that is enabling them in these inshore areas to head rapidly in the right direction. Sea trout, on the other hand, tend to stay near the river for a longer period of time. And again, on the west coast with these studies, it's been possible to have a look at impacts of seal predation with new methods. In this case, pit tags. These are devices that you'll probably all have now in your car keys, which tell the car that these are the right keys to stop the immobilizer. And we can actually put these inside these little fish, and we can put a detector, a pit tag detector, on the back of a seal down here, so that effectively when the seal eats one of these tagged smolts and digests the flesh and the pit tag goes through the guts, it will be picked up. This detector will relay the number of the smolt that it's detected, and this device uses satellites to get a position, and the seal will then basically phone in to say, this is where I am, and I've just eaten smolt number 325. So really, with modern techniques, it's remarkable what we can do. This has been developed for sea trout smolts, and that we think now that it should be possible to develop it for salmon also. And I have to thank Scottish Natural Heritage John Baxter, particularly for the efforts he put in with Sea Mammal Research and the Atlantic Salmon Trust and ourselves in developing this technology. Okay, so that's fine. On the west coast, fish can almost head in the wrong direction to get rapidly out. But it seems highly unlikely that the same rules would be used by salmon on the east coast. It's likely that a magnetic sense will be important. It's likely there will be local adaptation. Fish from different parts of the country will have different behavioral methods. And information is needed now urgently on the emigration behavior, particularly the vectors. We know that fish tend to swim at about a body length per second. What direction do they swim in? We have good models of currents. We should be able to combine these data and come up with a model of how fish find their way away from Scotland. So what about returning adults? What route should they follow? Well, if they hit shore, do they have some inbuilt sense map of how to get back to their river? Or maybe if they hit shore, they have to stay very close to the landmass to make sure that they don't lose it. Probably, if you imagine a fish that's been out at sea and first hits land, how's it to know just exactly where it is? It seems likely that it wouldn't even know which way to head, whether to turn left or right. And indeed, those data that are available from tagging studies, old tagging studies, suggest that pathways are much more erratic, and fish might have to search for a long time before they can locate the odour cues from their home river. So we would predict that adults, that there are complex searching behaviours, that they would use a range of depth as they look for odour cues, and they use a range of senses, compass and olfactory cues. Well, we started to have a look at some of these issues in satellite pop-up tags. And this is a satellite tag. It's really quite hefty. And I've brought it along out of interest, really, because it's actually a Scottish invention. And this one was built by Monty Pried in 1980 and was used to track basking sharks. It was a marvel of miniaturization in its day. It was incredible that it had been reduced from the size of a suitcase. And it's been a few years, but eventually we've got one that is small enough to go on the salmon. And it's been very exciting starting to collect data on homing salmon returning to the Scottish coastline using these satellite tags. And we've also started to look at the response of fish to electromagnetic fields. So I'll show you a few data from these early data from these studies. So the idea of these tags is once they're attached to the salmon, they collect depth, temperature, and light. And after a programmed time, they pop off the fish, they float to the surface, and they relay that information to satellite. So we have to catch the fish. We use a netting station off the north coast, and we work with the local bag net fishers. So the net is fished. Have a look to see if there's anything there. If there is, then the net's pulled in. There are the fish. And we can catch fish in very good condition. This is a sort of guess who the scientist is, for those of you who might know the individuals, and all will be revealed. Yes, it's Davy Stewart. And here he's releasing a satellite-tagged fish. And you can see the tag just on the back of the fish here. And off it swims, collecting data to relay back to us. Remarkably, this individual fish, just to give you an example of the whole process, was released on the 30th of May this year. And we knew that the tag had popped off on June the 1st. And so we had a trace of data that had been collected in this period. We don't know exactly where it was. We only have locations where it was tagged and where it popped off. But it was actually picked up again, the fish, minus the tag, of course, in the upper Tay. And indeed, this animal would have swum right past my window. It was really quite exciting that we'd gone up to tag it and it had actually come back again. So it's quite something. But if we look at, that was the fish I just showed there. This shows the distribution of pop-off locations that we had. So we tagged fish at Armadale here, and these fish were from quite a range of areas. or heading in a range of areas. And here we see some of the data. So these are bars of night, and this is depth. So the scale here runs down to about 90 meters, and you can see that this salmon was diving frequently over this period of time. And here we see the change in temperature. And temperature is quite important because by having temperature also, we could pick up when fish were eaten. So in this case, this fish dived down at this point and there was then picked up by a mammal. We know that because of the temperature, the body temperature. So actually this period relates to the diving behavior of the mammal that had eaten the tag. And these are the raw data. This is really quite an exciting track. This is light here in green, and it goes very dark at this point. And in this instance, the temperature goes up, not as high as a mammalian temperature, so we think it was eaten by an endothermic shark. And indeed, we got the tag back. So this is the tag that was inside a shark, and there's actually some little marks on there, which are probably tooth marks. Actually, this tag was used to track a basking shark. So one to track it and one eaten by sharks. But if we look at the depth distributions, there's quite a range. We've got 3 different fish here. This fish spent most of its time in the top 5 meters. This blue individual tended to use the water column much more frequently. And if we put all these tracks of fish together, we had data from 46 out of 50 tagged fish. We can see, if you like, the average pattern. And what we're finding is that fish generally, for most of the time, are in the top 5 meters of the water column. There's quite a lot of variation. All the individuals dive quite extensively. The maximum dives imply that the fish are going right to the bottom. And in terms of marine renewable devices, they're most likely to interact with surface devices, but could come across devices at any depth. So thinking about these surface devices then, I mean, a concern that has been brought up on many occasions is, well, what about the effect of magnetic fields from these devices? We've been having a look at this in an experimental facility. This is a 10-meter diameter annular tank. It extends across here. And in this case, we've got some coils that the fish voluntarily, quite happily swim through. But what they don't realize, of course, is that we can activate these coils and generate a field that is very similar to that from cabling that we would expect. What I'm going to show you is some salmon, and it's the first time that these coils have been energized. And we haven't finished fully analyzing the data, but it gives you a hint of the response of the fish to an AC electromagnetic field. So here's a fish approaching. This is a camera from above, so this is the same fish, and you can see it's approaching the coil. And there we go. There's another fish coming up now. And these fish just moving through this coil. Well, I don't want to prejudice our full analysis of the results, but it might be a struggle to find major effects. So how do we move forward? Well, we really need to understand a bit more about post-smolt distribution I was mentioning earlier. We're dusting down old particle tracking models at the moment. We're designing experiments for acoustic tracking of fish in the coastal zone. We are soon letting a project to look at timing of smolt distributions. And we're looking at camera trawling, the same sort of devices that Ken was talking about earlier, and getting these integrated with our standard pelagic trawling systems. And the other issue of importance is noise modelling. So these are all issues that are underway. And in terms of looking at adult behaviour, we're continuing satellite tracking work. We're looking at possibly acoustic tracking fish, hopefully looking at early deployed subsea generators, noise modelling, and developing better systems for looking at the population status of salmon. Finally, to thank all the researchers who've been involved. Thank you very much.
If there's an immediate question, we'll take it and then we'll move into the discussion. one back. Sorry, in the middle, 4 in.
Just a very simple question, but you probably, other people know the answer, but I wondered how, if you lose the tag on a salmon, you can follow where it is. How on earth did you manage to find where it was after it had lost the tag?
Yeah, I probably rushed through a little bit. The dash tracks are just the shortest route. We don't know where the fish is. All we have is the location where the tag was put on and the location where it pops off. We think if we tag a large number of fish, we will get a picture, if you like, along the lines that Ken was showing earlier. Is there a band following the Scottish coast? Are all the fish fairly close to the coastline? Because we'll have a lot of pop-off locations, but we can't track individual fish. It would be possible to put little acoustic transmitters on fish, but it's extremely difficult to stay in touch with the animals on the high seas. But it's a direction we might need to go in if the satellite tracking work doesn't give us the data that we need.
Thank you. Can I ask the speakers to come up to the front and we'll move into the general discussion. I've been asked by the Royal Society of Edinburgh to ask people who want to join in a discussion if they would wait until they've got the microphone, hold it close to their mouth, and keep their comments and questions short, please. Can I ask for the first person who would like to contribute? There's a lady 5 or 6 rows back.
Hi, my name's Hazel Hall. I spent most of my childhood sitting on banks watching my father fish. And something which he talked a lot about and hasn't come up today is salmon disease. Have you done any work on the disease that salmon get, that horrible sort of white yogurt-looking thing on their bodies?
Ken, would you like to start?
I think maybe John might know a bit more about that than I would, because it's part of the responsibility, I think, of his lab.
Sure, yeah. I mean, we have a disease section based at our laboratory in Aberdeen who sample wild fish routinely. And certainly if there are outbreaks of disease, they quickly having a look to see if there are any issues that can be dealt with. There have been 1 or 2 diseases. Red vents syndrome was a concern that arose quite recently, but that doesn't seem to be a sort of a terminal disease on the high seas. But I think you're right. There might be something that hasn't been picked up that could be a problem.
So the gentleman here.
It's Eric Salmon again. Judging by the length weight data, it suggests it's a feeding problem. Have they been doing surveys of the density and distribution of plankton in the Norwegian seas?
Yeah, I might take that one. Yes, in the context of SALSI, we tried to pull together all the data and the information that was available, and certainly in terms of the Salmon Summit, there's a couple of really good papers in that ICES journal that actually looks at the Sir Alistair Hardy Foundation data and correlates it then directly with the salmon. In summary, what we're finding really is that the planktonic organisms are moving north, and we're getting an exchange between the ones that originally lived in the south and the ones that originally lived around our coasts are moving further north. Some of them are moving at a rate of approximately 30 kilometres, about 18 miles a year. So the lower band of their distribution is moving north. There's not a gap, but what's happening is you have new creatures moving in. And if you look at some of the reports that we're getting at the moment in terms of the changes in the ocean in relation to particularly jellyfish, we have very major changes happening in the ocean as a result of this. So if you are a fish moving to sea and for hundreds if not thousands of years, your ancestors have been used to a particular pattern in terms of the actual feeding pattern, you may find it quite a shock when you go out there and find creatures that are either too big or too small, too nutritious or not nutritious enough. So we think it's all indicating change. And I think that really is reflected in Chris's results in terms of the thinner fish as well, is that the seas are going through these enormous changes, particularly that area in the southern Atlantic.
Are they doing stomach contents when they're catching?
Yes, we did extensive work on stomach contents in terms of the salmon and indeed the sister pelagic species that we looked at. Found some interesting differences between the two, but primarily they were pelagic feeders. They were in their smaller stage. The salmon tend to be quite near the surface. It's in their second year at sea that we started to pick up then this phenomenon of them diving, and certainly some of them dived to extraordinary depths. You know, some fish were going down 600-700 metres. We've no idea why. We're only beginning to try and understand that.
Peter.
Well, just to add a little bit to that, there have also been major changes in the quality of fish like capelin, which are a major prey of salmon. The lipid content of those fish, particularly in the Greenland area, I think has declined quite markedly.
Several people. Right at the back, there's a gentleman. Yes, there.
Thank you. John Gibb, Lochaber District Salmon Fishery Board. Long before we had fish farms or climate change, some people believed that the worst thing that ever happened to Atlantic salmon was the discovery of the feeding grounds in the North Atlantic. I'm just wondering, with all this really good work going on for all the right reasons, we're now potentially going to get a picture of all the migration routes all the way back to Scotland and other places, and all the way around the coast of Scotland, right into natal rivers. And I just wonder what the panel thought about the risks of that, of putting that information out there, bearing in mind we've got a legal and illegal netting industry going on. We've got very high value in the price of wild salmon at the moment. And Ken Whelan mentioned marine protected areas, and I just wonder whether it's ever been thought that there could be a move towards making sure that we protect what's found out through this work. I'd just be interested in the views of the panel. Thanks.
Goodness, who'd like to tackle that? John, would you like to say something about marine protected areas?
I don't know that there's much more we can say at the moment, Michael. I mean, there is a programme of work going on at the moment to identify marine protected areas, but salmon wasn't one of the special features for which areas could be identified at the moment. I think we probably need to know a lot more about exactly where the salmon go before we could go down that road. In terms of making this information available, we have something called the Freedom of Information Act, and I suspect that it would be very difficult to sort of hide this information if we were asked for it. But John might want to say something more on that.
Well, we certainly can't hide the information, I agree with that. But I think if you think about the situation with smolts on the East Coast, we really don't know how closely they follow the coast or head out. And I think it's imperative, if we're to advise sensibly on developments in the coastal zone, that we've got a knowledge of where the animals are. We can't protect them otherwise.
Peter, you wanted to...
Well, it was really just not so much in coastal waters, but of course IUU fishing is a big problem for fisheries globally. We did have some problems in the North Atlantic. It was mainly in the northern Norwegian Sea and in international waters. It was mainly involving vessels that were reflagged from Denmark to Poland and Panama, which are not signatories to our convention. But we were able to deal with that through diplomatic channels, and we haven't had any sightings in those areas since 1993. So I mean, the surveillance is not perfect, but it's pretty good. There are airborne surveillance flights. The Norwegians go over the area that was targeted because they police the Jan Mayen fishery zone. So we think we're getting a feel for it, and we don't think there's a problem now. With regard to marine protected areas, so far as targeted fisheries, legal targeted fisheries for salmon are concerned, they are prohibited by the convention beyond 12 nautical miles. So you have a huge marine protected area already.
There was a question or comment right at the back. Sorry, you wanted to.
My name's William Fraser. I was just, it's probably, you've answered it already. I was just wondering if there's any correlation between the salmon research and the skinny fish and the increase in the stock biomass for the mackerel and the the Atlanto-Scandian herring, and if there's any crossover research done between the two.
Sorry, if I understood, the question related to the abundance of mackerel in the Norwegian Sea.
Yeah, the increase in the stock biomass of the mackerel, western mackerel stock, which is increasing and moving into Icelandic waters.
Yeah.
And if there's any crossover research done between.
To be honest, the short answer is no, but I think there may well be a competition problem between post-smolt salmon and mackerel, especially once salmon have moved on to feeding on small forage fish. And my suspicion is that that's the key to understanding the changes in condition that we see, the climate-related patterns. My hunch is that it comes down, it's firstly the zooplankton which Ken referred to, but I think that the key thing is that switch to piscivory, and that's the high energy, high lipid, high growth potential diet for them. That's why they go to sea. Whether or not one would be able to actually draw connections with mackerel, herring, capelid, it starts to become extraordinarily complicated. I think in essence the fish are kind of filtering a lot of this information and giving you the end result. There may be several interacting factors which drive the final result. I think the other key thing which SALSI has offered, which my work cannot offer, is that by definition I'm looking at the survivors, the good guys. I have no idea what's happening to the ones that don't come back. Now, we could talk for hours about this, but it's a real problem with the kind of analyses that I'm running, and indeed in many cases where you really don't know where these things are going, what's happening to them. To give you one very brief thought, you could see an improvement in the condition factor of salmon finally returning to Scotland, but that might actually predicate serious deterioration in marine mortality, because basically only the very, very good guys survive to come back. So even if the quality of the return adults improves, in the absence of information on abundances and mortality rates, you can't use it as an indicator for a change in ocean conditions. And these are serious analytical problems.
Ken.
Thank you, Michael. Can you pretend for a moment that I'm a 6'7" Norwegian? And I'll try and give you the answer that Jens Christian Holst gave us some years ago at a conference we had run by the Atlantic Salmon Trust. Jens has a very clear theory at the moment in terms of exactly what's happening in the oceans. It's very interesting to look at what has happened in relation to the mackerel, the herring, because it follows directly along his predictions of 2 or 3 years ago. And as far as Jens is concerned, we're seeing a cyclical move in the oceans that has been repeated, as far as he's concerned, back to the 1790s and beyond. And this is a movement whereby you get a yin and a yang. You get a period of high productivity in one part of the ocean and low productivity in another. And interestingly, what we're finding is that in the western side of the Atlantic, it's going through a period of high productivity. The eastern Atlantic is going through a period of low productivity. Chris's work, as he said, is based around grilse. I was lucky enough on a couple of occasions recently to work in Greenland, and I have some absolutely remarkable photographs of the condition of the fish in Greenlandic waters. Now, the problem is that those fish that are in Greenland are obviously 2, 3, and maybe even 4 sea winter fish. So it's a very slow turnaround between them being there and them coming back. I would be very surprised if we looked at our really large multi-sea winter fish that were coming back if we didn't see a very major improvement in terms of the condition of those fish. So we've a really complicated situation here. We have natural cycles in the sea, and the tendency always at the moment is to look towards climate change and say, climate change, therefore that equals global warming, therefore man is influencing this. We're in a situation where man may be tweaking the actual effects that we're seeing in the ocean. But if we don't understand the actual cycles in the ocean themselves, we have very little hope, I think, of actually understanding exactly what is happening. I think that question in relation to the mackerel and the herring is fundamental to our understanding, because if we don't get into bed with the marine scientists in a much closer way than we've done heretofore, we haven't a hope of understanding this. It's one small pelagic fish out in that great big ocean. And just to give you 2 numbers, in the 1960s there was probably around 8 million salmon feeding at sea. There's now 3 million fish spread across that ocean. It's tiny, tiny number of fish. So if we're going to make any inroads in terms of understanding it, it has to be as part of understanding the pelagic community. I think that's a key point.
I think there's another interesting thing that came out earlier this evening. We're demonstrating a lot of correlations between things, but a correlation doesn't prove cause and effect. And I think your point about the link between the salmon people and the fisheries people, we need to know much more about the causes and effects and not just seeing these correlations.
Absolutely. I think that's fundamental. And if we don't understand what we're dealing with, and it really is a highly complex area and a changing area, don't understand what we're dealing with, there's a real risk that we'll get it wrong.
Yep. There was another question someone wanted down here. There's one at the back.
Hi, Chris, Ken. It's Katie Sumner from the Environment Agency. Have you, along those lines, looked at the links with the North Atlantic oscillation, particularly with the condition factors? Obviously, that's been bandied about in the past. Is that still going? Do we have any better idea?
I think so. Yes, Kate, the short answer is the North Atlantic oscillation is it's a very broad-scale index, summary index of very complex atmospheric and oceanic effects. That said, you can see broad patterns. It has quite a complex periodicity. There's at least 3 different cycles that underlie the periodicity of the NAO, ranging from a few years, 2 to 3 years, up to multi-decadal. It seems to be amplifying in recent years. It depends which index you use. Some people have used should I say, slightly unconventional NAO indices to track salmon responses. If you actually look at the standard accepted NAO index, which is relevant to this part of the Atlantic, there is no significant correlation. Excuse me. But I think that that's a reflection of the fact that it is you're condensing an enormous amount of information down to a single figure. And I think you lose a lot of the kind of resolution that you can gain from actually looking at ocean climate on a degree grid. But I think that excuse me, it's like the Atlantic multi-decadal oscillation is another very crude index of ocean climate, much longer periodicity. And people have tried to correlate abundance patterns with the AMO. In fact, I have done myself with other colleagues. But I think that they are very crude indices, and we lose the detail, the resolution.
John, does Marine Scotland get involved with these sort of indices at all? Or?
As long as Chris is doing it, we're quite happy, actually. I think that obviously a lot of what we've talked about this evening is very expensive, very demanding of resources. And I think actually, Ken's done a superb job with Peter in bringing a large resource to bear. And I think, as he mentioned, there's 4 years now of a gap. I guess we're trying to fill in some of the coastal areas, but we're very happy that Chris and the other university workers are looking at some of the detail.
Thank you. Question here.
Thanks very much. Paul Knight from the Salmon and Trout Association. That leads me on to a slightly, I think, the elephant in the room here, probably one for Ken, maybe for Peter. There are an awful lot of countries in the Northern Hemisphere with an interest in salmon. We've heard that we don't know enough about it in the high seas. We don't know enough what's happening in the coastal migrations. I would suggest we don't know enough of what happens in the estuaries as well, the transitional waters. Is there the political commitment across the Northern Hemisphere to find out more? Is a lot more being spent on our sea fisheries than our salmon? How do we fund Son of South Sea?
Ken.
Yeah, a simple question, really. But I would remind you that I got quite used to being called a lunatic in the early 2000s when we started off looking at this whole South Sea question. We hadn't a penny when we started, but we managed to put together a vision. And I think we managed to get buy-in terms of that vision. I don't think that vision is dead at this stage. I think it may have hit a hiatus, and obviously we've had the financial crisis across the world. But I think we really need to reinvigorate it. I'm fascinated by what technology can do for us now, particularly in terms of the massive amount of money that countries like Canada are putting into their ocean tracking networks. I think never again will we see a SALC programme where we look at salmon. I think we can forget that. We tried really hard to make sure that we had a programme that didn't just include salmon. We were thrown out right, left and centre. We were accused in Brussels at one stage of rebranding salmon so that we would get our hands on marine research money. And that puts it in context in the sense that it wasn't seen as a sea fish. It was, it really needed to be looked on as a freshwater fish, and people were comfortable with that because that's the way their administrations were managed. I think at this stage we've prized open that door, but we really do need now to be advocating the idea that the salmon needs to be wholly adopted as a pelagic fish. It needs to be discussed on an equal basis with the mackerel and herring we talked about earlier. That's not happening, and that's not science stuff. This is all to do with quasi-politics, and it's to do with discussion. And I think we need to mobilise that discussion, and I think we have the basis for putting the arguments forward. And in many ways, maybe we can make that gap work for us in terms of indicating to people, you've invested what is in essence nearly 6 million of EU funding, between the matching funding and the EU funding, into this programme. The EU are always very anxious to make sure that any funding that goes into major research programmes is not wasted. Well, at the moment, it's not far off that in terms of us moving it forward. So I think we need to rebrand it. I think we need to move it forward. And I think organisations such as our own, Paul, I think that really has to be our job, try and advocate for that and to get people interested in the idea at the ICES level and so on of looking on salmon as a monitor, this aquatic canary that can actually be useful to a whole range of pelagic species. And certainly in the context of the tracking workshop that we had recently looking at how to move this process forward, it was very clear that any tracking equipment that we're going to put at sea at enormous cost, it has to be a multi-species array. People are really interested in mammals, they're interested in whales and so on. If we can piggyback, no pun intended, on top of those bigger marine mammals, and the salmon is also an iconic species, we can build that vision again.
Peter, would you like to say anything further?
Well, I think Ken's covered it really well, actually, Michael. But I think from the point of view of fundraising, what we did find for SALC 1, whatever you care to call it, was that the fact that this programme had been put together by an intergovernmental commission, it was endorsed internationally, it had a lot of partners, it involved public and private sector, the NGOs, was extremely attractive to funders. And I think, as Ken said, the way we're looking to move this forward now, SALC 2 or whatever you care to call it, we're looking to partition mortality. We've got a lot more information on distribution and migration. What we're looking for now is quantitative estimates of survival, which we didn't have from the first programme. We think that means going down the route of tracking. We think there are some areas where there are choke points where we could have a raise if we can raise the money. But as Ken has said, I think it'll be absolutely crucial if we can involve some of those other iconic species in that network. I think then the chances of success in getting more money to take this forward will be much better. They've done it in the Pacific.
Sorry, we're going back to the elephant in the room again. When I leave this conference, I've learned a great deal, but my friends who are sports fishermen will be asking me, why do the West Coast rivers have such a fantastic drop in their population and the East Coast don't? They will say in the pub, it has to be to do with fish farms. And yet no one seems to go near that. I know it's about lost at sea, but surely, if there's a huge confounding difficulty about the transitional waters, why are you not giving us a hint as to whether the West Coast has gotten, particularly the Solway Firth, which has had a cataclysmic drop in its rod-caught salmon this year? They want to know why that's happening. Can you not give us some information?
Would anyone like to tackle that one?
Well, to an extent, it comes back to cause and effect and correlations. If one looks at sea trout around Scotland, they're doing very well in the Tweed district. They're doing very poorly in the Moray Firth, and they're doing poorly on large parts of the west coast. It's likely that these fish are experiencing really quite different conditions in these different parts of Scotland. Salmon the same, and trying to work out why the trends are different in different areas is not straightforward. I think some of the work that Chris has been involved with, for example, where one actually does some experimental type of work to try and test exactly what the causes might be. For example, protecting smolts against sea lice are stronger in terms of inferring effects. I'm sure that doesn't help your friends in the pub. But
I think one of my surprises this evening has been that there's been so little mention in the talks about genetical aspects. When I looked at, for example, Chris's different groups of fast, slow growing, I would love to know if there are genetic differences in them. Is it just because it's too expensive, too difficult, or why haven't we heard much about the genetics of salmon?
Well, number one, I think it was probably to do with time constraints, to be honest. But certainly, we have Eric Verspor, I see, in the audience, and Eric is really the leading light in this regard, and hopefully we'll hear a good deal more about it over the next 2 days in Glasgow. But I think the genetic breakthroughs that have been made as a consequence of SALSI will far outlive SALSI itself and the results of SALSI. It has opened up the possibility of having a toolbox whereby we can actually start to differentiate and separate out the various stocks of fish. I think we're just on the cusp of that at the moment. It's relatively expensive. It's not that expensive. It's becoming far cheaper. And I think it's a matter of people focusing in and making that a priority, if you like, in terms of a requirement in relation to research objectives. I think the technology is there now, and I think it certainly can help us and can do an awful lot in terms of discriminating all of those very fuzzy areas that we were talking about tonight.
Could I just also make one other comment? The data I was showing you with these different growth patterns pertain to mixed stock fisheries, which we don't know which river. We could, I guess, from the scale samples, we could DNA type them with your SNP markers, but it's time and money. But one thing I can offer, which is, I think, not irrelevant, is that when we look at the River North Esk as a single river, we see exactly the same mix of patterns. So it's not a river-specific pattern that we're seeing. It's a response of fish of multiple genetic composition, and I think it's one of these situations where it's the final result of the integration of a lot of information. But there may well be genetic differences which underlie these, and it would be very simple to map that information onto genetic information. But it's like everything else. It's time, money, and also energy, which I'm losing as I get older.
Well, when we hear of time, money and energy, I think it's time to invite Lord Nixon to propose a vote of thanks. Lord Nixon.
Thank you, Michael. Well, I have the easiest task tonight, but I have to thank a great many people. First of all, I'd like to thank John Alexander and the Royal Society for hosting this tonight and for the initiative in allowing this forum to take place. And secondly, to his embarrassment, Michael Usher for chairing the committee that organised it so efficiently and sticking to his timetable. I don't know how he did it, but well done. And also the staff of RSE who have organised this so efficiently. And it's very nice that Laura Dean, who has been responsible for this and hasn't been very well, is back with us tonight. My brief was to thank the speakers. You won't want me to thank them individually, but all I would say is that anybody who has ever stood on their feet will know that for a 20-minute slot you have to put in 20 to 30 hours or a great deal more and a lifetime's experience in preparing the slides and preparing what they've said. So all our speakers, thank you very, very much indeed. And apart from for the RSE, who have financially put this, I should also thank some sponsors. The Fishmongers Company, Colin Bogue, who Clark is here, and we'd like you to convey our thanks for your contribution to this conference and to so much else that Fishmongers have done through Andrew Wallace in Scotland and continue to do. And also SNH, who helped financially, and the Cromarty Firth Fisheries Trust. Simon McKelvey, the director, is here. That's just one of 20 to 30 fisheries trusts that work throughout Scotland so closely with the fishery boards. And finally, thanks to you, the audience, and also particularly perhaps to the overspill audience who haven't had the fun of sitting here with us tonight and actually seeing our speakers face to face, and to say that there won't be a person in this room or outside it in the overspill who does not feel passionately about this wonderful species. Otherwise you wouldn't be here, and there must be hundreds of thousands of people who would like to be here too. And it just demonstrates how vitally important it is that we all continue to work and support our knowledgeable scientists and NASCO and everything everybody's doing to try to get to the bottom of this and hope once more to see salmon in the abundance that our forefathers remembered. Thank you all very much indeed.